I just want to start by putting everybody on the same page in terms of the way I think of utilizing molecular markers. One of the first ways that we can use molecular markers is to identify high-risk populations or people that are at risk of developing cancer in so-called molecular epidemiology, an area that I think is very, very important, which is taking early lesions of any type, whether it be in the preneoplastic area of head and neck cancer, colon cancer, et cetera, and identifying those lesions or genetic changes that are likely to produce the highest risk lesions that progress to cancer. And of course, the area that we're all very, very much interested in, which is in patients that already have cancer, what are the molecular markers that can predict the best therapy? And I'll spend most of my time, I'll provide a couple of examples of each of these in the area of individualized, personalized therapy. Just to take everybody to the same page, cancer is caused by the accumulation of genetic and epigenetic changes. Genetic alterations include point mutations, deletions of chromosomes, et cetera. And we're going to focus today on epigenetic changes, which are a little bit different to some of the main markers we're going to talk about. And epigenetic alterations just means that it's not the DNA basis, but something beyond that. For example, methylation of cytosines or histone modifications all fall into epigenetic alterations. You'll see, based on the assays that we've developed, why we're targeting specifically promoter methylation in the development of some of these markers. Just to bring it back again one more level, this is a genetic mutation. Once a gene has an allele has a mutation and develops an advantage, the cell develops a clonal advantage and begins to outgrow other cells, it usually loses a second allele. In promoter methylation, this is the promoter of a gene. This is the cytosines that populate this promoter. And you can see here that over time, you can actually methylate or add methyl groups to these cytosines. As it gets denser, you get less RNA off this gene promoter, less and less protein. And in fact, when you have dense methylation, which is what we detect with our quantitative assays, you get essentially no protein and the equivalent of a gene inactivation event that you would see from a genetic hit. This is the basic assay that we use. We take DNA from any source, so this can be from a primary tumor, from blood, from urine. We then treat it with bisulfite. You see here I've put the methyl group on the cytosine. If the cytosine is methylated after treatment with bisulfite, it remains as a cytosine. If the cytosine is not methylated, it chemically is converted to a uracil. So then you have essentially two strands of DNA, either the non-methylated strand, which now has U's, or the methylated strand, which has C's. You then do PCR with primers that detect either the C's or the U's, and you use an internal probe that can then detect, again, additional C's or U's internally to this gene promoter, to then both with real-time PCR and fluorescence, you can actually see the signal for specific methylated alleles, and also it's quantitative. You can tell how many methylated alleles you have. I just want to talk very briefly first about early detection, how to use methylation markers in the early detection of cancer. And we focused here on urine and in the detection of bladder cancer. This is an old painting from the 17th century looking at urine. I think we've become a little bit more sophisticated now in that we can extract things like DNA and protein from the urine for analysis. So our first approach at using these methylation markers in bladder cancer was to take the kinds of patients that walk into institutes like Rambam that are tertiary care patients and just look at their urine. Most of them, as you see, out of 175 patients had transitional cell carcinoma, then various others that you would see that are the small minor mix that make up predominantly invasive bladder cancer. There are, again, about a third of lower grade, and most of them are higher grade, which is what you usually see. Some, again, the mixture between invasive and non-invasive. One of the things we've learned over the years, and this is one of the mistakes that many people make still in the development of biomarkers, is that you need an appropriate control group. So we take our controls from the same hospital-based population, again, a nice mix here of males and females, patients that have various confounding situations. So these aren't just normal postdocs that you can grab out of the laboratory and take their urine, that are also 30 years younger than your population. These are people of the same age, and again, they have the kinds of problems that are likely to be diagnostic dilemmas. So for example, atypical cells by urine cytology and all these other things that we have here are all the kinds of things that are likely to confound the diagnosis of bladder cancer. And many of these patients are likely, for example, to have blood in the urine, which again is is a common manifestation of many diseases, including bladder cancer. Again, not looking at the gory details of all the methylation, but just to look at simple comparisons, what I have here is simply 15 patients, just to show you the first 15 that went through the system. These are the gene targets that we're going to look at their promoters to see if they're methylated or not. There are genes here that you may have heard of, like P16, the gene on chromosome 9, that is inactivated early by promoter methylation in many types of cancers. And next to it, ARF, which is also right next to the gene and is also an important tumor suppressor genes. And then some other genes that don't sound so familiar, including genes, again, like APC, which are mutated in colon cancer and methylated in several tumors as well. What I want to show you here is this is tumor, and this is urine. So these are match cases where we have the tumor specimen and the urine specimen. And when we see methylation in the urine, we always see it in the tumor. Here, for example, again, or here, et cetera. So this just shows us, again, that it makes sense, that we're doing essentially what's called analytical validation to make sure that what we're seeing in the urine is, in fact, what's coming from the tumor. There are situations, though, for example, where the tumor has methylation, we don't see it in the urine. So that's essentially a failure of the test. We assume that, again, because there are methylated alleles in the tumor, they should be in the urine, but that's not always the case. Either there's not enough cells or enough DNA that's being sloughed off into the material. But again, the specificity is going to be high because you only see predominantly methylation in the urine when it is present in the tumor. This next slide is the way that I like to show these biomarkers now is scatter plots. I think they're the best way to show you and give you a feel for the type of information you're getting from these studies. In addition to that, it also gives you a sense of the range when it's a quantitative assay such as the ones we use, and brings us more to the older types of protein assays as well that were very quantitative. So just want to show you that these are 4 of the top genes that we found. Now remember, we can't really plot all the zeros here for these close to 100 patients are all down here, so we don't do it to really crowd the graph. But you can just see that there's just a few positive for APC methylation, while most of the clinical samples that have cancer are positive, but you can see the range. Some very similar to controls or close to 0, but the majority higher. E-cadherin, for example, also very nice, and just a few positives. A couple that look almost perfect, ARF, not quite as common in the cases, but again, it's absolutely no controls have methylation of ARF, and no controls have methylation of GST1 right here. So those, again, would be very specific markers. One of the challenges we have is how to bring these tests into the clinical scenario, and we have to move from what's called analytical analysis or analytical sensitivity, which I showed you here, which is just looking at tumor and urine to see if it makes sense that the event is present in the tumor and the matched urine, to the issue of clinical sensitivity, where we don't really care necessarily what the status of the tumor is, and we may not even have tumor. Our analysis there is, does the patient have the disease? Do they have bladder cancer, or do they not? And then we assess the clinical sensitivity and specificity by simply what happens when we test the urine. So here we increase the number of samples to all the samples that we had, 175 cases, and again to the 96 controls. And you can see here that we have 4 perfect markers. We have nice sensitivities for each marker between 28, 35, 43, and 45%, while all of them have 100% specificity. Remember, these are the ones I showed you where none of the controls were positive. And then there's these other markers which are not bad with various sensitivities as well. The specificity is not perfect because you saw there was a few dots on the scatter plot, and you can actually develop cutoffs then, which are still very low, but you want to get your best point on the receiver operating curve. Now, of course, it suggests that you have a good marker, but how are you going to combine them to be able to use them in the clinical setting to maximize sensitivity and specificity? So we developed these receiver operating curves, which are simply plotting sensitivity up in the y-axis versus 1 minus specificity, or which looks at false positives on the x-axis. And you're really looking for a curve that goes up as high as it can up to here. Here would be a perfect test at 100% sensitivity and specificity. This is the 50% flip a coin type of situation, which of course is not what you want, and anything better than that is good. You can see here that these markers are pretty sensitive and specific, and then they kind of drop off individually as you try to go a little bit farther up. Again, so individually they're not bad markers, but again, you don't get into the very high areas of the curve. So the point is, how can you combine these markers to try to get the best point on the receiver operating curve? You don't have to look at this. It's just to show you how complicated that can get. You can use all kinds of combinatorial approaches to try to figure out how to combine your markers. What I like as a clinician scientist is that they all kind of give you the same result. which is that at approximately 80% sensitivity, you can get specificities in the mid-90s. So that makes me feel good that no matter how you analyze the data, the markers are kind of giving you the same type of information. The only difference is whether you use 4 or 5 markers together, how you combine them, et cetera. Just want to point out that the likelihood ratios, which usually more than 10 means that it's a fantastic marker, are very, very good here as a combination. And the likelihood ratio, the negative likelihood ratio, which is its specificity of less than 1, is also very good. And you can see here 0.15, which means that as combinations, these are excellent markers. Another thing that we've liked to do now in my group is using all these different approaches is plot the best point off the receiver operating curve. So this is just the top part of that ROC curve that I showed you before. So it's just from the 80% to 100% specificity. And again, just the top part of the sensitivity curve. And you can see here that no matter how you combine markers, they all fall within what I call a cloud, which is simply, again, the numbers are not that different no matter how you combine them. But it does serve to be able to see how one marker, for example, can push it up a little bit or push it down. The nice thing about this type of approach that I think is very useful is that when you bring in one new marker, you don't have to repeat everything all over again. You can just see how it changes this curve up or down to see if you can maximize, again, your sensitivity or specificity. And this is an area you're going to see a lot more of as we begin to combine these biomarkers. One other interesting thing, of course, is that these aren't just markers. They're actually epigenetic changes drive the progression of cancer. So as you would expect, you can also see what kinds of correlations you see with simple clinical pathologic parameters. These are cytology. This means that if in these cases they were identified as positive under the microscope through cytology, which means that these cells had a morphologic change that allowed you to see them as cancer cells compared to those that were negative. And you can see here 2 markers cluster very nice together on the dendrogram, showing that these methylation changes, when they occur, cluster together and also identify, again, this morphologic change under the microscope. And 3 markers that cluster together have all been previously identified as invasion and late-stage markers, and again, they identified those patients as well. So one of the other values of using markers that are also part of the progression pathway is they may not only identify cases with cancer, but also tell you something about them in the clinical pathologic parameters that you're interested in as well. So going from early detection to this issue of, again, therapeutic prediction, I want to take you through just a few steps in the laboratory, and the rest will be very, very clinical, in terms of how we try to identify markers that can identify either a sensitive population to a particular therapy or resistant population. And by the way, this is something that is very, very tough to do because it's hard to identify things in the laboratory, usually cell lines, though they can be other types of models as well, as I'm going to go into that are sensitive versus resistant. And even if you do, when you identify these or create these, you then have to jump into the population. And this has been the biggest problem in terms of developing markers and going all the way to the population, is first not only their identification, but in this crucial issue of validating which of the patients, in fact, are going to be those that are predicted to respond to an individual therapy. This is just a quick look at at oncogene mutations that may have some relevance in the clinic right now. We really don't have a lot of time to go into them, but I just want to point out a couple of things. Everybody knows that EGFR mutations in a small percentage of lung cancer patients could be very important to predict response to Tarceva. I'm going to get back to this issue of KRAS specifically in colon cancer and sensitivity to Erbitux. I've heard already that this is an issue in Israel that is being considered in terms of national testing for patients with colon cancer. I just want to mention that BRAF is intriguing because BRAF-specific inhibitors were just reported at ASCO with a very, very high response rate in both melanoma and thyroid with a specific Plexicon product that just inactivates the BRAF mutation, and PI3K inhibitors are also coming to the clinic. Again, in addition to these oncogene mutations, I talked about promoter methylation, and the reason that we're looking into this area is there already is another very good example, in addition to those oncogene mutations, of a methylation event that predicts sensitivity and resistance. And those of you that are involved in treating brain tumors know it very, very well. Gliomas that do not have methylation of a gene called MGMT, which is a repair gene, are unable to remove the bulky adducts that are formed upon treatment with alkylating agents, and so therefore the tumor becomes resistant. Those that are methylated no longer have the enzyme and again become sensitive. This study was published in New England Journal of Medicine. It actually came out of another group from Hopkins. But again, it lends a lot of support to the notion that some of these methylation markers can be very important in predicting resistance or sensitivity. This list is here. You can read it. This is one of the things that we have to deal with in the laboratory, from the identification of a pathway to identification of the tools that are necessary to look at sensitivity and resistance, to then identifying the biomarker, doing a technology assessment to see if you can actually get that marker so that it can test things that you want to test, like tissue samples or bodily fluids, retrospective analysis that have to be done to see if your marker really identifies what you think it does or not, and then eventually prospective clinical analysis, only at the end to still be cut short if you can't offer it in a commercial setting. So while many people think that developing biomarkers is a lot easier than developing a drug, I just want to point out that it in many ways is the same and potentially even harder, because there's often less financial backing to develop a marker versus a therapy. So this is one approach that we took, looking at markers that could potentially predict sensitivity or resistance to a very common agent that we use in the clinic, cisplatin. Obviously, the main therapy that we use in lung cancer and head and neck cancer, it's certainly useful also in bladder cancer. So our approach was to basically take 3 sets of cell lines that had been engineered over a period of time, where they originally were sensitive to platinum, then over a year or 2 became resistant to platinum by treating them with low doses of platinum. We actually got them from 3 different institutions in 3 different continents, so we thought that if there was any bias, we could take that out by looking at all different cell lines from different places. And then what we do is we can treat cell lines with 5-azacitidine to reverse the methylation. We get re-expression. We can then start selecting which genes are common among these cell lines. We can look at the literature to cut it down, actually eventually identify those that are truly methylated, and eventually try to show, and this is a crucial step, not only that the gene predicts potentially sensitivity or resistance, so that's just a marker, but then by actually doing RNAi knockdown to show that it's part of the process of developing the cisplatin resistance. I'll just show you very quickly that this is what 5-azacitidine looks like. I'll show you one gene we're going to talk about. So for example, here's the parental cell line. This is KB. Here's the resistant cell line. Again, if you treat with 5-As in the parental cell line, nothing happens. It's highly expressed in both. But here you have a gene that's been turned down, presumably by methylation, in the resistant cell line, and then when you treat with 5-azacitidine, it comes back up. And this is the way we do, again, these differential displays to try and identify genes that are methylated in the resistant cell lines. We then have to prove it. So one of the ways you can do it is just take your DNA, treat it with bisulfite, and then you sequence. And of course, remember I just told you earlier that if it's methylated, if the cytosines are methylated, they remain as Cs, and if not, they turn into uracils. So you can look for either As or Cs. And again, you can see here that in the resistant cell lines, the As have become Gs over here because they have become methylated. So this is just a list of some of these genes that we had. These are 14 genes that looked interesting. These are the 3 different cell lines, again, sensitive and resistant. And again, just looking at whether in the sensitive cell line or the resistant cell line, they're methylated. You can see here that these are genes that are predominantly methylated in the resistant cell lines, and the ones that we're going to be looking at specifically as important biomarkers. Now, one of the things I told you is that we want to make sure that they, in fact, are not just markers, but potentially are the cause of the resistance. These are RNAi knockdowns. So, of course, now we have the ability to make these small molecule RNAs that you can put into a cell and knock down the gene, so we can recreate that same situation of methylation. Instead of turning off the gene at the promoter methylation, which we cannot do, we can take a cell line that has a gene that's being expressed, take out the RNA, and essentially, again, reduce its expression to the point that it's similar to producing methylation. And again, what you see here is when we do this with SAT with 2 of these genes with the siRNA, we can make them resistant. Again, this is not much for one gene. This is pretty good here for one of the genes to knock it up, about 30% resistance. And part of the issue here is it seems not one single gene can produce the entire resistant phenotype, but a combination of genes come pretty close to producing, again, the resistant phenotype. One of the things you can do when you move from cell lines, moving down the process, is to move to xenografts. I'm going to talk a lot more about these xenografts or models, but these are lung cancer xenografts that we have. Again, here, this is 1, 2, 3, 4, 5, 6, 7 of these. We have 3 that are responsive to cisplatin. I'm going to show you why this is so interesting, because these aren't just xenografts. These are derived primarily from patient material that are still, and these patients are still alive. And you can actually, and we know that they're platinum responsive in the clinic, not just in the mouse. And then again, we have those that are non-responsive. You can look here at our genes. the 4 that we thought were the best. You can see here that, again, you can just glance here and see that your methylated genes are much more predominant in the non-responsive or resistant tumors. And there's only one tumor which seems to come out of whack with 2 genes that are methylated. And you can see here that if you look at the methylation index, you can already start quickly identifying those tumors that are likely to be more resistant. So again, that's one step before the humans, but again, with good human data to begin to validate these genes. And we also got kind of lucky because as we came out with one of these important markers, S100, we found out that while we identified it in our own panels, it had been for the last 20 years identified as a gene whose expression was decreased, especially potentially in tumors that had a platinum resistance. And in this very interesting study, which was published in 2007, we saw already that S100 expression just goes down dramatically after tumors are treated in the neoadjuvant setting. So you take a tumor, you treat it with platinum, and what happens very quickly, within 2 or 3 months, you can see the S100 expression drop dramatically. This was an independent study done by another group. But what's interesting, of course, is that it shows you that the most likely scenario, which we've just shown, is that this is due to the accumulation of promoter methylation, downregulation of the gene, and therefore, again, the production eventually of resistant disease. So just to summarize that quick data, we've identified some important methylation markers. And the other issue for a common treatment like platinum, but the other issue, which is very important, of course, is that we've shown you that we can also produce some of the resistant phenotype by knocking down the gene, suggesting that it's not also a marker, but also a part of the development of the resistance. Now, I want to spend the rest of the time talking to you about something that I think is really transformational in oncology, and that is the development of these new types of models that we call tumor grafting. To get there, I just want to show you a little bit of what's been going on in the field of development of models for therapeutic assessment. And to make a long story short, again, it's very similar to that slide where I showed you. This is just a diagram. You have to start with a molecular target, do things in cell lines, find out the MTD in mice, the maximum tolerated dose, and then eventually move to this thing called human tumor xenografts, which most of you know is just cell lines that have been grown in the laboratory. You can inject them into the flank of a mouse. They grow as tumors, and you can test various drugs. Now, I just want to point out that it's probably not a good idea that this happened. The xenografts were originally identified or produced actually in the early 70s, and they really were produced as continuous cell lines so that you could send them to different laboratories across the world so that people could get these cell lines, grow them in mice, and do their sensitivity testing wherever they were. There are already certain things that were known in the 70s and 80s, which I think remain big challenges. One of them is that cell line testing in general is only good for cytotoxic agents. So in general, the best thing you get out of cell lines, if you just do simple death assays, is obviously whether cells live or die. And again, the most important agents there are some of the major damaging agents that were developed in the late 70s and 80s. There generally is not a great prediction for sensitivity. Even in the best studies, they said 50%, which means that just because something works in a cell line or a xenograft doesn't mean it's going to work in the clinic. And we know that very well because only 8% of drugs that are moved forward by the pharmaceutical industry ever succeed in the clinic, even though they had potentially great xenograft results. There is a notion that at least cell lines are okay at predicting resistance, at least for damaging agents, and that makes sense. If you throw in a damaging agent in high dose in tissue culture and the cell doesn't die, it's very unlikely that it's going to die again once you put it in as a xenograft in the mouse. And there are other things as well, but that we don't have time to get into. This is a xenograft here in the nude mouse, just to show you. And again, this is what's been used traditionally. And one of our approaches that we took was based on actually older studies that were done in Germany in the early 80s, where somebody had the notion that instead of growing cell lines, maybe they could take a piece of tumor directly from a patient that's still a fragment or a solid piece and implant it into the flank of a mouse or into the organ and see if it would grow and could be continued to grow and continue to grow as, again, a solid fragment. This is actually a study that was done showing sensitivity and resistance to platinum. These are some of the common cell lines that have been used, and here's 28-day TC. This is what you measure in vivo in xenografts. This is the IC50 in cell lines. And you can see here that for platinum, for example, you have a pretty good correlation. It's one of the things that led people to say that, you know what, it doesn't matter whether it's a cell line or a xenograft, the data is going to looks pretty good. The mistake was that once you move away again from these damaging agents, the data was not as good. In these tumor grafts originally established, they saw very good correlation, but they were very hard to propagate. What we do now is we basically take a piece of tumor from any given organ that's affected with cancer. We implant it in what's called the engraftment phase. So you take the piece of tumor, you put it into the flank of the mouse. Then when it gets to be about 3 or 4 centimeters, we then sacrifice the mouse, cut the tumor into various pieces, which are then put into several other mice. This is the first expansion phase. By this point, and we have now about a 90% take rate for these tumors, you know that the tumor is going to survive. By the way, the time from surgery to implantation for maximal results has to be about 2 hours, which is one of the limitations in terms of getting these to grow. Once it's expanded, we then expand it out through 4 or 5 generations. We now have hundreds or thousands of mice available from which we do our pivotal studies. And I'm going to show you some very interesting things about this model, including the fact that they don't change over time during the time that we have to change them to sensitivity and resistance. And then I'll show you, I think it's much more predictive for drug screening as well. So the first thing, if I'm going to tell you this model is better, I have to tell you that I have to at least show you some molecular data. If you take a standard xenograft and you run it on a 30,000 gene expression array, there's only a 20 to 25% similarity between the primary tumor and the standard xenograft, which is pretty amazing when you think that the entire drug development field is based on using these xenografts. If you look at a primary tumor and a tumor graft, there's a 94% correlation. That means all the genes are going in the same direction, except for a few, and about half of those are the angiogenic genes, which are coming from the mouse, which are not on the array. So at least on a general gross molecular point of view, they look pretty good in terms of similarity. The other thing you want to see at is what about sensitivity and resistance to various drugs. So here I just want to show you between passage 5 and 10. This is, again, just looking at tumor volumes. These are tumors that are decreasing in size. These are tumors that are increasing in size. These are resistant. These are sensitive. You can see here that all the graphs are very similar, plus or minus 20%. They're all going in the same direction between different passages. One single exception here, that's 0 versus 20. But again, for the most part, these are they remain very, very similar from passage to passage in terms of sensitivity or resistance to various drugs. At the gene level, we can show you that they don't change. This is RAS mutation. This is a group of pancreatic tumors that have been established like this. You can see here the specific mutations established at F0, which is essentially the primary tumor, and at F3, and again, you can see no change in the mutation status. The sole exception here is this pancreatic tumor graft here, where it goes from mutated in one allele and wild type in the other to mutated in both alleles, probably a selection process, but again, not a change in the mutation that takes place. In addition to genes, we want to look at proteins. For pancreatic cancer, we're interested in a signaling, downstream signaling protein of TGF-beta, DPC4. Again, between passage 0 and 3, no change in the expression pattern, except in this pancreatic tumor graft, where it's a little heterogeneous in its stain, and then it goes to essentially homogeneous in its stain, but again, in the same direction. What about a response to a larger number of tumors? So I think this is one of the most pivotal slides that I'm going to show you. If you were to take, for example, 30 patients with pancreatic cancer and give them gemcitabine, you'd expect in the clinic to have about a 20% response rate. So it's very interesting because if you take 30 of these tumor grafts that are derived from 30 patients again, and you treat them with gemcitabine, you see two very interesting things. First of all, you see over here the tumor graft study, which is here in the larger green and red. Again, you see the sensitive tumors over here, which is about 20%, which is what you would expect in the clinic. So it's predicting the same response rate that you're seeing by treating these tumor grafts that you would see in the clinic. And again, the resistant, completely resistant tumors. And then this right here are actually RECIST criteria of the actual patients, very closely mirroring the results that we saw in the tumor grafts, identifying those patients that by RECIST criteria had some response and those that, again, did not have any response or were resistant. Then you can take it one step further and say, okay, does response actually correlate with time to progression or survival? So this is actually looking now at the tumor graft data. So again, looking at the same 30 patients that received gemcitabine in the adjuvant setting. And what's very interesting is you can see those that do well, which means that their tumor grafts show a response, had a survival that was nearly 2 times different in terms of the median number of days than those that did not have a response or did poorly in the tumor graft testing. These graphs that I'm showing you here are almost precise reproductions of the actual phase 2 adjuvant setting data that was actually shown to be the case in real trials. So what I'm trying to get at is that not just individually are they correlating with responses, but you're also seeing in phase 2 data that you can predict that response rate very well. One more, Tarceva. Some of you may know that Tarceva has been approved in combination treatment with gemcitabine for pancreatic cancer, a whopping 11-day difference in overall survival between the combination versus single agent alone. The response rate as a single agent is probably about 10% if you give it to patients, again, if you give Tarceva to patients with pancreatic tumor. And it's very interesting because those that had some sensitivity out of these 30 that we tested, again, was just 10%. So you can take these tumor grafts and predict the same response rate using Tarceva here that you did for gemcitabine, which again gives you the notion that you can start using groups of these tumors to actually predict what's going to happen in phase 2. So what about individual patients? This is a patient with lung cancer, a non-small cell lung cancer patient that was resistant to platinum. He was actually the CEO of a cable company in the New Jersey area. He actually paid for his tumor graft to be implanted so that he would have some sensitivity and resistance data. And what's very interesting is he failed first-line therapy with a doublet, which in our case is usually platinum plus a taxane. Failed second-line therapy with Alimta. And then we got these results, and you can see that the tumors already start as established tumors. And you can see here that you keep essentially a straight line up to 63 days. This patient, this graph over here shows irinotecan, sorafenib, and Avastin, something you would almost never use in non-small cell lung cancer. He was treated with this because really at third-line therapy there was very little else to offer him. He had a 50% maximal remission, a durable remission for 9 months, and actually ended up doing well for about another year after that. Here's another patient with a leiomyosarcoma, 55-year-old female. This one I think boggles the mind because she was very interesting. She had the leiomyosarcoma, had surgery, and then after surgery had adjuvant therapy with gemcitabine and Taxotere. So we would normally not go back to that therapy. She had a 6-month window after adjuvant therapy, which she had before she recurred. And then she had 2 other lines of therapy before, again, we had the data in the mouse. Now here's day 78, and you can see here how flat this line is. Essentially, the tumor just doesn't grow. And this, amazingly enough, is gemcitabine and docetaxel again, plus Avastin. And this is without Avastin. So they're pretty close, but with Avastin it's a little bit better. So again, in a situation now in 4th-line therapy that we normally would not go back to the adjuvant situation, we give her these drugs. She goes into a 50% maximal remission for 9 months and actually responded to another therapy line that was predicted from this approach afterwards. And again, in 6th line, which would be almost impossible to see normally in a sarcoma. This is another sarcoma patient. This one was just published last year in Cancer Research. A 45-year-old male with a mesenchymal chondrosarcoma, a very rare type of sarcoma. What was interesting here is not only that we identified drugs that could work, again, that kept this tumor flatline, but at the point that we wanted to treat the patient, again, this was 3rd line, he had multiple metastases of the sarcoma throughout the bone, pancytopenia, it was platelet transfusion dependent, blood transfusion dependent, white count of 0.2 with low-grade fevers. We were very scared to treat him with anything, even though we had a curve here that suggested that treatment with irinotecan and docetaxel or docetaxel and Avastin would really suppress the tumor. And we actually just gave him the docetaxel on a weekly basis, and from the first dose of docetaxel, all his counts went up, and he ended up living another 18 months after this without being transfusion dependent at all because he had such a nice response. This is one of our most dramatic patients, a pancreatic cancer patient, which was actually one of our first academic patients treated in the pancreatic tumor group. And it's very interesting because when we looked at this particular tumor, you could see here about a 50% growth reduction for gemcitabine, and somebody pulled mitomycin C off the shelf, a very severe damaging agent which is not used too often, and you can see here that essentially had no growth. The patient went on to be treated with gemcitabine, which is the standard. CA19-9 was about even at about 20,000, and this is a typical response, or really just stabilization of disease. Spiked to over 100,000, was then treated with mitomycin C, and after 6 cycles went to 0. The patient actually went into a remission for 2 years, recurred, and was treated with another drug that we predicted from this approach, platinum, and did very, very well. And in fact, is still in the remission. This tumor is particularly interesting. I just want to show you here on the clinical situation. These are the multiple Mets that you can see, and this is after treatment with mitomycin C, a completely clean liver. Very unusual for metastatic pancreatic cancer. This patient was actually one of those that was completely sequenced at Hopkins. It was part of the whole genome project, so it had complete sequencing of all the genes. And we found out that the gene that was mutated here was the Fanconi N gene, which is part of the Fanconi complex, which is why I think this tumor was so sensitive to DNA damaging agents. So here you have a very nice genotype-phenotype correlation in this particular case. Here's another patient with gastroesophageal cancer. This patient now went through 3 lines of therapy before this tumor graft grew. You can see here a very nice flat line for something that we might have thought of. Irinotecan, cetuximab, and Avastin is right here. And the reason it's only out to 18 days, you can see how fast this tumor is growing. This tumor was doubling probably every 20 days in the patient and in the mouse. He went on to respond very well with a 50% decrease in the CEA, felt much better. A lot of his liver lesions resolved. And actually, this was between April of 2008 to December of 2008, and then he recurred again. The CEA hit 1,000. And the patient was actually from London. There was a lot of resistance to treat him with anything else. But we had this data. This is Abraxane and Avastin down here. And the other thing that was very interesting, you can see here you have established tumors, because remember, they're tumor pieces. So in the mouse, it starts at 250, and you can see that it's actually going down over time. 5 out of the 5 tumors in this case were going into a remission, and we really pushed him to treat him with Abraxane and Avastin. His CEA levels now since December are back to normal. The patient is feeling quite well, and these are again his scans. This is lung metastases now, which have been completely resolved after therapy. Now this is 6th-line therapy for metastatic esophageal cancer. You can see most of the liver lesions have completely disappeared with a small lesion left over here. So the patient continues to do very well. In fact, wants us to do more testing, see if we can come up with more drugs that might be effective. And lastly, I just want to go over one more individual case, which I think is quite fascinating. This is a 38-year-old female with stage 4 metastatic colorectal cancer. It was very interesting because we really were very limited here in terms of financial resources. And so instead of these 20, 30, 40 drug regimens, which I showed you, we only tested 7. And what we did is we started with the standard regimens in colorectal cancer. So we did FOLFIRI, we did FOLFOX, we did all the combinations in the mouse. And then we found out that the FOLFIRI group was doing the best of all, and the FOLFIRI group was really producing straight lines. But the issue was we had time, because even after she recurred, she still had only minimal disease. So we had time to dissect this out. And the interesting thing, which I think is also fascinating, she received FOLFIRI originally as her adjuvant setting 2.5 years before we were ready to treat her at this point with recurrent disease. So we were interested to see which of the drugs within the FOLFIRI regimen she was likely to be benefiting from in this tumor graft studies. And amazingly enough, we dissected it out and found that irinotecan as a single agent produced this line. For those of you that can't see it, this is day 56. And again, you're starting with an established tumor, and everybody can see here that this is a real remission. And again, in 5 out of the 5 mice that are used in the group, all 5 of them went into a remission. All those tumors shrunk to 0. And we felt very confident that irinotecan was the only agent that she had remained sensitive to from her original adjuvant situation. By the time this was done, she actually was not doing well. The small amount of disease she had grown. She had massive pelvic disease the size of a basketball, multiple lung mets. And in fact, there was a big argument about whether to treat her at all or put her into hospice. Given this data, she was down in Alabama, we really pushed the oncologists there to treat her, and they refused. So she went to another oncology practice. And she was treated with single-agent irinotecan. And what I asked them to do is just to give her a little bit more dose intensity with irinotecan, to give it to her every 2 weeks, just 3 cycles, and let's just do the experiment. And after 6 weeks of irinotecan, she had a 60 to 70% reduction in the size of this pelvic mass. She had bilateral hydronephrosis, which completely resolved. She pulled out her PICC line. She went with her son to Disney World, and in fact, now is in a complete remission after only 12 weeks of single-agent irinotecan. So again, in multiple lines of therapy, we've seen these to be predictive, one patient after another. This is just a summary of what I've shown you in lung, leiomyosarcoma, the other chondrosarcoma, gastroesophageal cancer, colorectal cancer, and pancreatic cancer. These individual patients, how many drug or drug combinations we used, the fact that they predicted clinical sensitivity every time, sometimes multiple times. For example, in the gastroesophageal cancer patient, 3 times, 3 times for this pancreatic cancer patient. And we've always predicted clinical resistance as well in the cases where we can. I mean, normally you wouldn't want to go to use the drugs that are resistant in your model, but sometimes retrospectively we can see that the patient did receive the drug and was in fact resistant. And we're expanding this now. There's about 40 patients under active treatment, so I think this list will continue to grow over time. But this is the kind of the single patient data just to add to that phase 2 data and the larger number of patients we've done with pancreatic cancer. And I just want to end by combining the way I started with the end, which is we're looking for markers that can predict sensitivity or resistance. One of the advantages of having such a model, and one of the reasons we're so excited about the prediction that they're giving us, is that instead of trying to go into populations and getting tissue from the pathologist to look at this marker or that, if you believe that the model is so predictive, you can do all your assays in that model. You can do your gene expression, oncogene mutation analysis. I already told you some of these tumors have been completely sequenced. You can do promoter methylation analysis, look at microRNAs, look at all your favorite biomarkers, and then just test them in the mice with the notion or the hope that everything you're seeing in this model is actually what you would see in the clinic, and eventually, of course, develop a biomarker that can predict sensitivity. So again, these are your tumor graft studies. Again, you're looking at various tumor grafts from various patients. You look at those that are sensitive versus those that are resistant. You do all your molecular assays that you need, identify the potential marker, and then hopefully just go once into your clinical trial testing with a lot of confidence that your marker is likely to be very predictive. So I just want to end with something I began with in looking at this issue. We talked about KRAS and the fact that it's so important now for Erbitux. And so you might ask, well, what if we had this type of approach before all this started and we finally figured out in the clinic that KRAS mutation could predict sensitivity or resistance? So I just want to show you, these are the pivotal studies based on KRAS status in response to cetuximab in metastatic colorectal cancer. The study started in 2006, didn't pay a lot of attention, 30 patients. 65% of the patients with wild-type KRAS responded, none of the mutants. But then, the data began to grow through 2007. By ASCO of 2008, everybody was up in arms. All the studies were showing the same things. You almost never had any mutant patients responding. High response rates of 30 to 50% in those that were wild-type. And eventually, the two crucial studies that were finally done, especially this larger study of 226, which very clearly showed, again, the very same results. I mean, it's very hard to find this type of clinical data where virtually every single patient that's wild-type are those that are going to respond, and virtually every patient that's mutant does not have a response. So you'd say, okay, what if you went back and took these tumor grafts from patients with colorectal cancer that were mutant or wild-type? It was interesting because I was working with ImClone at the time, and they did not even know the KRAS status of the Xenografts that were used in the first clinical testing to take the drug to market. So we took standard tumor grafts that we had grown from patients and looked at KRAS versus mutant versus wild-type. And here, a very simple combination of cetuximab plus oxaliplatin gives you a completely flat line. And again, if you're mutant over here, you have absolutely no response. So you could at least say that if we would have used the tumor graft testing, not only would have been able to identify sensitive and resistant tumors, but because they would have been characterized, we could have already seen that it was likely that the KRAS mutant patients would be resistant to the therapy. And this, I think, is the pivotal study that finally showed this to be correct. This is the absolute best experiment you could do in the clinic. You take patients that are KRAS wild-type with metastatic colorectal cancer, and you give them Erbitux, or you give them nothing. So this is essentially just one drug by itself versus the mutation status. This is a study that was done that showed very nicely a very large difference in survival in the wild-type patients with zero difference in survival, zero, between getting nothing or getting Erbitux if you're mutant. I mean, one of the most definitive studies that has ever been done. This is actually a large Canadian study that was sponsored by ImClone. And I want to just tell you that if you want to measure KRAS, so one of these issues that again, came up actually in our conversation about what's the best way to do it, we've developed a simple assay, which is a colorimetric assay, which can quickly identify mutations versus background. I just want to show you why it's so important. We've also published, again, going back to this issue of biomarkers, that you can detect circulating DNA from tumors in the blood. So what happens if you take patients now, instead of having to necessarily go and get tissue, what if you can just get plasma and just see if you can see circulating DNA from patients that are either KRAS, wild-type, or mutant, so that you could identify those patients that are likely to respond to Erbitux. And the data is pretty dramatic. These are colon cancers with mutant KRAS, and you can see here they identify them all very, very nicely in the plasma. We can identify about 70% of patients rapidly just by taking a ml of plasma and identifying the circulating mutation. And these are colon cancers that are wild-type with one exception. Again, we don't know that's a heterogeneous tumor. There's another mutation or could potentially be a false positive. But not a bad test when you have to think about the fact that you can draw it when the patient comes to see you in clinic rather than trying to find out who has the tumor block and how many cells are there and all the other stuff that you have to go through. So again, just very rapidly, this is 72% sensitive and 93% specific. And just a very simple colorimetric assay. By the way, it costs $8 to run. It's one of the reasons we like it. It's not very fancy, but it's very, very effective. And again, to tie this all together, this is the reason why it's so important. You can get a rapid, reliable DNA test. You can do it instead of a biopsy. And of course, in this particular disease, bringing back, again, the issue of models and prediction, what you have is a patient where you don't know the status. On average, if you give Erbitux to all these patients without knowing their KRAS status, there's an average improvement in survival of about 8 weeks. If it's wild type, it's 6 months. If it's mutant, it's 0. So if you ever wanted a situation in the clinic where you wanted to know that if you wanted to give the patient an expensive drug or not, this is the best situation we've ever seen where a single biomarker can accurately distinguish between these two groups. And again, very, very easy to measure. So as a summary, tumor grafts, which we've been developing, have the ability to study key signaling pathways and identify targets and signatures. I think it's a unique platform to correlate biomarkers with response prospectively, where you have a lot of confidence that what you're seeing in this model is likely what you're going to see in the clinic. This is probably one of the most exciting parts about this. Could you run a mini phase 2 study in mice before you run it in humans? So you could predict your response to single drugs or whether a drug has to be combined. And then you could also power your study in the phase 2 setting correctly by studying the tumor grafts before spending all the money in the clinical situation. The other thing, of course, which is very interesting, is what if the drug doesn't work very well? What if it's something like Tarceva, where you only see a 10% response rate, but you identify those patients that are likely to respond based on biomarkers? Then you could actually do a phase 2 study where you pre-select your patients based on the biomarker, let's say a 2-to-1 sampling. You put patients into the study predominantly to have the biomarker, then you can increase your response rate and actually accelerate your drug approval by doing that, by identifying the biomarker that can select the best population. So tumors arise through the accumulation of genetic and epigenetic changes. The oncogenes drive the cancer process, and some of those are important that we've talked about, like EGFR and KRAS. The tumor graft models can help predict sensitivity and correlate with patient responses. I think we can glean biomarkers in a much more productive fashion than just going straight from cell lines into a clinical population. But I guess one of the interesting things is this is early. We're only treating about 40 patients right now in these tumor graft studies. We have a lot more data on the pancreatic side, but obviously it all needs to be eventually validated prospectively. I just want to tell you that, again, as Carl mentioned, there's a lot of people that deserve all the credit. All these people in yellow are the ones that work on methylation. Manuela Hidalgo helped develop this tumor graft model, especially in pancreatic tumors, and are our clinicians that help this become possible. Thank you very much.